So let's see, this is our principal investigator who researches on this muddy effluent, toxic muddy effluent which is dumped in this river. And you are an enthusiastic PhD student who decide to spend 5 prime years of his life with this bald guy. And the project is kind of very simple. You wish to isolate as many bacteria from this spot of this muddy effluent, and you want to see how many of the bacteria that you isolate from this spot uh, have gene 1, 2, and 3. And you have already designed primers for gene 1, 2, and 3. All you want to do is isolate bacteria, do PCR analysis, and see whether they have amplify gene 1, 2, and 3, and see whether they have it or not. Right, so it's kind of simple. You created a grid in Excel sheet where, let's say, you have isolated three bacteria: one, bacteria 2, bacteria 3, and you want to amplify gene 1 in bacteria 1 using primer specific for gene 1. Similarly, gene 2 in bacteria 1 with primer specific for gene 2, likewise gene 3. So you are already looking at 9 such tubes. You also need to generate a no template control. Basically, what it means is that it will have all the components of a PCR assay, like magnesium chloride, tag buffer, tag polymerase, and all those things, but it won't have the, the DNA template. Let's say it won't have the bacterial genome, right? Um, if what that will tell us is, in case of no template control, if you still find that there is amplification, then you know that there is some contamination of bacterial genome in one of the reagents of PCR assay, right? So similarly, you also have a positive control. Now, what if you didn't get any amplification, and you doubt that maybe one of the components of PCR assay is not working? Let's say your tag polymerase is not working. So for that, what people typically do is that they have a positive control. Let's say they have a gene of which codes for a flagella protein, and they have already cloned it into a plasmid and purified that plasmid. So now they use that as a positive control, and they have designed, let's say you've designed primers for this flagella gene. um, so every time it works and it gives you an amplification. Now if your positive control is working but your is not working, then you know that one of the components, one of the reagents which you're using for PCR assay, there is some problem there, right? So um, in one of the previous tutorials, we have already seen how to assemble a PCR assay. So PCR assay will have magnesium chloride, tag buffer, nucleotide mix, upstream and downstream primers, tag polymerase, template DNA, nuclease free water, right? So for, let's say, for gene 1, this is the assay system which you've already established. You add 2 microliters of magnesium chloride, 1 of buffer, 1 of nucleotide mix, 1 microliter each for upstream downstream primers, 0.25 microliters of tag polymerase, 5 microliters of template DNA, and nuclease free water, right? So you make the entire assay system of 50 microliters. The same way you have also developed assay systems for gene 2 and gene 3. And so usually, so all the components will be similar, and very similar amounts of or the aliquots of different components you will add in all three assay systems. They may be also similar, identical also, but one of the things would be very different: upstream and downstream primer, right? Because these are three different genes, of course, like they'll have three different primer pairs, and based on different, if you are adding different amounts of these components, then of course you will be adding different amounts of nuclease free water, so that you can make up the volume to 50 microliters. So let's see how to assemble a whole uh, assay system for all these three different genes. So let's say you decided you want to uh, take a 96 well plate, and you would use each well of it as a tube, and you also generated an Excel sheet. So let's say cell 1A will have bacteria 1 genome, and you are going to create an assay system to amplify gene 1 with the PCR primers of gene 1. Similarly, bacteria 1 gene 2, bacteria 1 gene 3, likewise bacteria 2 gene 1. So the first row entirely contains the test PCR assays, right? It will test whether these bacteria amplify for gene 1, 2, and 3, and do they have these copies or not. It's just a hypothetical experiment. Whereas in row 2, you have the no template control for bacteria 1, 2, 3, no enzyme control, where you have the entire assay system except for the enzyme. And then you will have a positive control, right? So let's say you have an E. coli genome, or maybe that plasmid what we talked about, which contains the flagella gene, and you have the the primer pairs. And so you may so typically you don't start assembling one tube at a time. It's not that you will take you will take magnesium chloride, then water, then nucleotide mix, upstream downstream primers, one tube at a time. Now imagine how much amount of time you will spend just assembling these 18 tubes or 18 wells, and how many tips you are going to waste for each reagent every time you will dispose of the tips, and a lot of plastic waste. So instead of that, and to reduce amount of time, of course, like you'll have a lot of pipetting error, right? I mean, you are dealing with 1 to 5 microliters amount, 0.25 microliters. So if you add that low volumes of liquid, there will be a lot of pipetting error. There can be a lot of variation from tube to tube. So to avoid all that, people create master mixes. So what do I mean by that? So let's say we created an Excel sheet, which the first panel mimics pretty much what we saw. You have test bacteria 1, 2, 3, bacteria 1, bacteria 2, bacteria 3, and you want to amplify gene 1, 2, 3 here. So all those 9 combinations of wells. Then you have no template control for bacteria 1, 2, and 3, no enzyme control, and positive control. Now, if you see all these tubes, and if you see the components of PCR assay, let's say magnesium chloride. So you're going to add magnesium chloride in pretty much all the tubes. Tag buffer, all the tubes. They'll have common, like PCR nucleotide mix, all the tubes. Upstream, downstream primers. So that's not common. There are 3 different genes. Uh, all 3 of them will have different pairs of upstream and downstream primers. So you'll have to add them separately. But if you want to make a single mix, you can probably add magnesium chloride, tag buffer, nucleotide mix, etc., right, to dispense. Okay, let me clarify it. I'll take a little bit more time. Then you have template DNA. It'll be different for for 3 different bacteria, so different genomes of bacteria. And of course, in no template control, you are not going to add any template, any DNA. Then tag polymerase, yes, except for in no enzyme control. Water, yes, you are going to add in every tube. So pretty much you can make master mixes. Like you can have, I mean, the whole idea is that instead of adding magnesium chloride, tag buffer, PCR nucleotide mix, tag polymerase, and water separately in each tube, when you know that you are going to add it in each one of those tubes. So why add them separately? Why not just mix all of them together and make a sort of a solution, and then just dispense one time. So in one go you are going to add magnesium chloride, tag buffer, all these things in just one go. So that way you are going to reduce the amount of time you take to assemble the entire assay. But it's not that simple. So like you are seeing here that upstream downstream primers are not common. So you are for separating components which you have to add separately in each tube, and or like each different set of tubes, and components which are sort of common, so you can add You can mix them together, make a master mix, and then just add in one go. So what I would do is here, like, I would prepare probably three master mixes. So I'll have master mix one, which will be good for all the test and positive control tubes, right? All the test and positive control tubes. Um, so here, in these tubes, we are adding the template DNA. We are adding all the components of PCR, right? In master mix 2, which is no template control, here we are not adding any template, right? Any DNA. And so there is, in case of nucleotide nuclease free water, sorry, in case of template DNA, it will be a 0 here, right? And for master mix 3, we'll have no enzyme control. So in this case, we won't have a tag DNA. So for example, for tag DNA, we'll just put a 0 here. So that way these three master mixes are kind of different. um, Now, for the sake of making our life easier, we just imagine that in all the for example, if you want to assemble PCR assay system for bacteria 1, 2 and 3, or let's say for gene 1, 2 and 3, let's imagine that you are adding equivalent amounts of magnesium chloride nucleotide mix, um, forward and reverse primers, let's say 1 microliter each or 2 microliters each, in case of all three genes, gene 1, 2, 3, right? So I mean, I'm just trying to make it simpler in the sense that uh, because all the components you are adding in equivalent amounts, you are also adding equal amounts of nucleus free water, right? So let's just try and do it, and that way it will become more clear. So let's say for positive control, we have 9 wells here, 3 wells each for every bacteria. Let's say bacteria 1, amplifying 3 genes, gene 1, 2, 3. Similarly for bacteria 2, bacteria 3. But for positive control, right, we'll just use 3 different assay systems, different primer pairs. So for gene 1, 2 and 3, let's say we are using E. coli DNA as our DNA template, and we know that it always amplifies those 3 genes. So we have 9 plus 3 tubes, right? So we have 12 tubes. So I'll write here for master mix 1, total number of cells 12, right? So this is excluding no template control and no enzyme control. Now what I'll do in the next row, I'll write total cells for calculation. So this is the total actual number of cells that I want to use. But for calculation purpose and to assemble the assay, the master mix, I'll use total number of actual cells plus 3. That means I'm calculating the entire all the liquids for 3 extra wells. And what's the reason for that? See, every time when you are adding liquid, there is some amount of pipetting error. You may be adding little less amount of solution or little less amount of reagent every time, right? Second is when you are using plasticware, and if you are dealing with very low volumes of liquid, many times liquid just sticks to the walls of plasticware, and eventually for the last tube you will find that your volume is little less than what it actually should be. Let's say you are dispensing 45 microliters in each tube, and if you are going on for 12 tubes, so till 11 tubes there won't be any problem, but for the 12th tube, instead of 45 microliters, all you would be left with would be maybe let's say 35 microliters. So to avoid that situation, what you do is you calculate for, instead of 12 tubes, you calculate for 15 tubes, right? So you add little extra liquid, little extra amount of each reagent, so that you don't end up with in that sort of situation, right? So you just add, like, whatever is the total number of cells plus 3. I mean, that's up to you, plus 3 plus 4 plus 2, whatever. Then let's say you are adding 2 microliters of magnesium chloride for each, in each PCR assay system, whether it's for gene 1, 2 or 3, right? So 2, and so here it will be total number of cells for calculations into magnesium chloride per tube, right? So that is 2. So 15 to 15 twos are 30. So now we have 30 microliters of MgCl2. Let's say you are adding 10 microliters of Taq polymerase buffer in each tube. Now there are 15 tubes, so 15 into 10, that will give you 150. Same way for DNTPs, right? So if you are using 1 microliter of, let's say, the nucleotide mix, so 1 into 15, that will give you I mean, the reason why you should embed calculations in Excel tubes is like, if you are going to use this assay day in and day out, every time, you will use it in different permutation combination. Sometimes the gene numbers would increase, sometimes the bacteria, kind of bacteria, the number of bacteria would increase. So all you need to do is, simply you change the number here. So instead of 12, let's say you put 20. So you see, all the number changes. So you don't have to break your head doing calculations every time when you start the entire PCR assay, right? Now, upstream and downstream primers. So these are going to be different for each of the genes. So let's say you are going to add 2 microliters each of upstream and downstream primer. So you're going to add it differently, I mean separately. Uh, Now let's say tag DNA polymerase, so 0.25 microliters. So of course 15 is the number of tubes into 0.25, that'll give you 3.75. So in one go just add 3.75 microliters of tag polymerase. Template DNA you're going to add separately. Nucleus free water, you are adding 31.75 microliters in each tube. So of course that multiplied by 15 gives you 476.25. So total volume is 675. Now divide 675 by 15. That is, this is the total volume that you have to dispense in total number of tubes, that is 15. So you get that as G21, that is 675 divided by 15, that is 45 microliters. So for a 50 microliter assay system, what you are doing here is you are adding 30 microliters of MgCl2, 150 microliters of PCR buffer, 50 microliters of DNTPs, 3.75 microliters of Taq DNA polymerase, 476.25 microliters of nucleus free water. Total volume is this, and from that entire master mix you will simply take a pipette pen and set it to 45 microliters and simply add aliquot 45 microliters in 12 tubes, right? So you will have little extra left, but you will add it into 12 tubes. Now each tube has gotten 45 microliters. To make the entire assay system of 50 microliters, you are still going to add 5 more microliters of something. Of that something, you will add 2 microliters of upstream primer, 2 microliters of downstream primer, and 1 microliter of template DNA, right? So this is where the rest of the 5 microliters will come, and that's how your entire assay system would be complete. So to add upstream and downstream primers, also you can make a mini master mix. Let's say you want it for 15 tubes, right? So 15 to your 30. So just take 30 microliters of upstream primer, 30 microliters of downstream primer, make a small solution, 60 microliters, and from that in each tube add 4 microliters, right? 2 microliters of upstream, 2 microliters of downstream, and that way you can add, you will save time. Similarly for template DNA, you pretty much can't do much. So you just add one microliter in each tube. And same way you assemble your master mix for no template control and no enzyme control, and then aliquot them quickly, and that way you will save time, right? Uh, so let's say you assembled the master mixes, and now you are ready to aliquot your stuff and start the PCR. So let's say you first do it for your bacteria 1, and you will just aliquot the master mix 1 for bacteria 1, right? So you will just simply take the pipette pen multi-channel, and you can put it for bacteria 1. Actually you can do it for because for all the tests it is the same thing. So you should just do it in one go for all 3, bacteria 1, 2 and 3, and not separated like what I'm showing here. Same way you do it for your uh, uh, for all. So that way, this is how you generate your master mixes and save your time to assemble the PCR assays, especially if the experiments are kind of more intensive in terms. So once you prepare your entire plate, then you sort of spin it down. Just give a brief spin so that all the components are sort of mixed nicely, and then you simply put it in your thermocycler. If you don't have that many wells, like here we are not using actually that many wells, just 18 wells, so you don't want to waste a 96 well plate. So you have such tubes, which are like strips of micro ependophs, my strips of PCR tubes, and uh, yeah, they are sort of connected, so you can assemble your assays in those small tubes, put them in microfuge and spin it, and do your PCR. So that way, just how this is how you, if you make your master mixes carefully, you save a lot of time in setting up your PCR experiments.